root parasites. But you also have parasitic weeds that are stem parasites. Look at hollow parasitic weeds that are fully dependent on their host for all of their life cycle stages. And they have no leaves and no leaf chlorophyll. And so they're completely white and therefore they're completely dependent on their host. An example of this is the Orobanche broomrape species. So you have different species of broomrapes, but they are within the genus of Orobanche. You also have obligate hemiparasitic weeds. So they're half parasites. They are fully dependent on their host in the below ground stages of the life cycle. But once they emerge above ground, because they have leaf chlorophyll, they can also sustain themselves for at least part of their nutritional needs, carbon needs. And a good example of this is Striga, which is shown on this picture in a sorghum crop. And then you have facultative hemiparasitic weeds, and I put the word hemi, half, here in between parentheses, because it goes without saying that if it is facultative, it cannot be a full, it cannot be a hollow parasite. So this type of parasitic plants, they are not dependent on the host for their growth, for germination and growth, but they can benefit through parasitism. So obviously these parasitic plants, they have leaf chlorophyll, and an example of this is Rhamphicarpa, and that is the weed that I'm going to talk about in this talk. So on this picture you see Rhamphicarpa within a rice crop, photographed from above, and although the Rhamphicarpa plants are a little bit reddish here, in earlier plants, in earlier stages of their life cycle, they are green, and so they do photosynthesize. Like I said, other types of parasitic plants are stem parasites. So the other distinctions are stem parasites and root parasites. But and then you can also distinguish parasitic plants based on what type of host they parasitize on woody species, on dicots or monocots or omnivores. But we will be talking mostly about parasitic weeds of cereal crop production systems. This is what I have been working on for most of my career. And these are all root parasites. So I have worked on Striga hermonthica, which is called giant witchweed. And this is an obligate root hemiparasite. Below you can see a distribution map of the parasitic weed in Africa. I also worked and still work on Striga asiatica. I also still work on Striga hermonthica. Striga asiatica is called red witchweed or also Asian witchweed, and it is also like Striga hermonthica, it is an obligate root hemiparasite. And then Rhamphicarpa fistulosa, called rice vampireweed, is a facultative root parasite. And here you see the distribution map and also a photo of the flowers when they are flowering, and they are only flowering at night. So they're only opening at night. So this photo was taken during the night. All of these parasitic weeds have similar hosts: maize, sorghum, millet, and rice. I put rice here in bold because the talk will be mainly on rice. And also because rice is the main host, at least for Rhamphicarpa fistulosa. And Striga is mainly found in maize and sorghum and millet, but also very much in rice, which many people are not really aware of. And then there's also sugarcane as a non-cereal, but also grass type crop that is affected by these parasitic weeds. So, like I said, this talk will be about Rhamphicarpa fistulosa. And this is a picture of the plant, flower and seed capsule. And I will be talking about the research that I have done on this parasitic weed in the period from 2005 till 2006 to 2023, so up till the current, and I'm still working on it. Before I started working on Rhamphicarpa, this is what we knew about Rhamphicarpa fistulosa. In 1835, the first taxonomic description was published on this species. In 1933, so almost 100 years later, the first published note on Rhamphicarpa fistulosa in rice was published, and it was in Madagascar. And in 1949 was the first published note on Rhamphicarpa in rice in West Africa. In 1975, a taxonomic revision was published, and also it came along an ecological and geographical ecological description and a geographical distribution. And then in the late 90s of last century, there were papers on ecology and biology. But since then, there was no research, active research on Rhamphicarpa. So this part of what we know is what we knew in 2006 was based on these publications. And so, for instance, you can see how the parasite looks like in the early stages, the 4-day-old seedlings and 7-day-old seedlings, and also a microscopic image of the haustorium, that is the connecting organ that connects the parasite to the host root, and that you can see here on the right-hand side in detail. And also Hansen published the distribution map of the species, and interestingly, this species is distributed in Africa and in northern tropical Australia, but there are no records of any of these of specimen of Rhamphicarpa fistulosa in between. So there's no connection between these different distribution areas, which is quite interesting. There are 5 other Rhamphicarpa species worldwide. And 2 of them are in Africa, but Rhamphicarpa fistulosa is the only known important and widespread weed. The other 2 are more found in Central Africa, and they are not known to be a weed problem. And based on the publications before 2006, we could sense that there was that it was an slightly increasing weed problem in inland valley rice in West Africa. Inland valleys are sort of landscapes where depressions where you have slopes and valley bottoms where rice is grown. So it is mainly rain-fed lowland rice. So in areas where water cannot be controlled, that is where this parasitic weed really thrives. So you can imagine that when I started working on this parasitic plant, there were many questions. It was like I felt like a kid in a candy store, because there were so many questions to embark on. For instance, how important is it? Is it invasive? How is the regional spread? Which countries are affected? What are the yield losses? What are the production losses, economic losses? How do the parasite-host interactions? How do they look like? And how important is a host for that parasite, because it is facultative? So to what extent and how does it affect its host? What are the preconditions for growth? The seed germination, the environment, and very important, because of course I am an agronomist, like I think many of you as well. What can be done against it? How can we control it? Is there varietal resistance, tolerance? Can it be controlled by cultural measures? What is the role of fertilizers? So all this I started to investigate together with collaborators and students. So I started working on this species in 2006, as I mentioned already, and this is when I was based in Benin, in West Africa. Sometime later, I moved to Senegal, still with the same research institute, Africa Rice Center, And I discovered that it was a problem there as well. And a few years later again, I moved to Tanzania, to East Africa, and there I also observed it to be a major weed problem. A major weed problem, but many colleagues, many weed scientists in those countries where I worked did not really know about it. And then I also worked in Uganda and in Madagascar while still being based in Tanzania. And also there I found it to be a weed problem that was relatively unnoticed, again, mainly by researchers I spoke to. Of course, the farmers, they were nevertheless very much affected and aware of it, but they didn't know what to do against it. So these are just a few pictures of the different countries that I just mentioned, and these are all infested rice fields. So sometimes it gets a little bit of a brownish, reddish color, as you can see specifically in the middle pictures. The left-hand side are the pictures from West Africa, and there it is. It's less. It turns less reddish than in East Africa. That's just an observation that I've made. So when I was still in Benin, I repeated a survey that was done before by Galbert Behunu, a partner, a colleague of mine from the national system. And they did a survey in 1998 in different inland valleys within Benin. And then we repeated that about 9 years later in 2007. And so we found in the same inland valleys that were surveyed in 1998, we found among those inland valleys, we found in 2007 that there were inland valleys that were newly infested, that were not infested before in 1988. So that was a clear indication that indeed this parasitic weed was spreading. Now in 2016, we published this study in Agriculture, Ecosystems and Environment. We really wanted to know what is the importance of this parasitic weed, because I found that it was quite widespread, but to what extent? And so we collected a lot of public herbarium specimen, and we mapped that and we overlapped that with rain-fed rice area data. We used literature data from the literature, and then we modeled this to come to incidence percentages and also economic loss estimates. So we found that this parasitic plant is it's very widespread, is occurring in 36 countries within Africa. 28 of these countries, they grow rain-fed lowland rice. So it is really from Senegal to Madagascar, as I've already shown you, and extending into southern Africa. And we estimated the annual losses to be above 200,000 ton annually of paddy, so unmilled rice, and that equates to around $82 million. And we also estimated that the total area invested should be around 450,000 hectares. And no surprise, because these are rain-fed lowland areas, so rice growing areas where water is not controlled. This is mainly a problem for smallholder farmers. So these are just two other maps representing the same herbarium specimen with the black dots, and then overlapped with our estimates of their incidence on the left-hand side. So how many? What is the percentage of inland valleys or rain-fed lowland area that we estimate to be invested by this parasitic plant? And on the right-hand side, you can see the production losses because of this parasitic plant, also differentiated per country. So this gives a little bit better insight in, okay, which countries are really most affected. And so you see Senegal, Burkina Faso, Nigeria, Mali, in West Africa, and Tanzania and Madagascar in East Africa, and also Uganda. The gray areas is where are countries that do not really have rain-fed lowland rice, or not where rain-fed lowland rice is not really important. Now we also studied the ecology. We wanted to know. Of course, we had seen it basically in rain-fed lowland rice, but we wanted to confirm whether this species has any ecological plasticity, and whether it was really restricted to rain-fed lowland rice. So conveniently, in Tanzania, and here you see two maps of Tanzania in the lower part below these photos. You see two maps of Tanzania. On the left-hand side is a map with distribution of Striga species. The green dots are Striga hermonthica, the brown dots are Striga asiatica, and on the right-hand side you see the same map with Rhamphicarpa fistulosa. And so in the south of Tanzania, that is where we found both species, and there is an area in Kyela district where both species are problematic, and sometimes farmers are affected by both species, especially those farms that have fields in different parts of the upland lowland continuum. So on the left hand side you see an aerial photo, and this is an area where you see Striga asiatica closer to the road in the let's say in the higher parts of this picture, and Rhamphicarpa in the lower parts of this picture, closer to the river. So all these squares that you see there, those are all rice fields. So we laid out three transects, and along those transects we sampled all the rice fields that we came across. And sorry. And this is what we observed in 2012 and in 2013. So we did this 2 years, these transects, and the red bars indicate where we found Striga, and also how important they are in terms of how much percent, what are the cover classes, and they are seen below this graph. And the gray greenish bars, they represent the Rhamphicarpa. So in 2012. There was really no overlap, and it was really a clear distinction. The Striga was mainly found in the higher and drier parts, and the Rhamphicarpa was mainly found or only found in the lower and wetter parts. But in 2013, which was a little bit drier, no, I should say there was a little bit wetter actually. We saw that the two ecologies moved closer to one another. So the habitats were more, there were actually fields, neighboring fields, where we found Striga asiatica and Rhamphicarpa. And there was even one field, and that you see that in the middle of this graph, in transect 2 in 2013, where there was one field where both parasitic plants were found. So it really depends a little bit on the rainfall, but in general, the two ecologies of these parasitic weeds are really separate. And controlled experiments, pot experiments with different water regimes, soil moisture conditions, they confirmed these observations. Now then we looked at the host effect. How do these parasitic plants affect their host? On this picture, on this graph, you see at 42, 78, and 111 days, you can see the host dry matter partitioning, and then you can see that for 0 Rhamphicarpa plants with 6 Rhamphicarpa Rhamphicarpa plants and 13 Rhamphicarpa plants. So you can see right from 42 days onwards, there are significant effects on the roots and on the stem, but not yet on the leaves. Also at 78, it's the same. And then only at 111 days, you can see significant effects on the leaves, and you can also see significant effects on the panicles. So progressive parasite effects, but rather late. The effects are rather late compared to what we know with Striga. And then on the right-hand side, you can see a picture where we had the green leaf area duration on the x-axis and the total dry weight of the host plant on the y-axis. And then for Rhamphicarpa free plants, and those are the black dots, and Rhamphicarpa infested rice plants, and those are the open symbols. And here you can see that even with increasing green leaf area duration for the rice infected plants, after some time, whereas the green leaf area is continuing, you see that the total dry weight of the plant is not continuing. So for healthy plants, you see a more or less linear increase. And for the Rhamphicarpa infected plants, you see that it is bending off, that it is flattening off. So this means that actually eventually the host plant, while still having green leaves, the green leaf area of the host plant does not benefit the production, the total dry weight production of the host plant. So in fact, the host plant is actually only producing assimilates solely for the parasite. So this is why we came up with this title of this paper, Slavery in Plants, because the Rhamphicarpa is really becoming a sort of a slave, a master, and rice is really serving only the parasite. So this is another paper where we have shown how it is affecting the host plant physiology. And then you see this is showing a time course of the photosynthesis light response curves for infected plants compared to Rhamphicarpa free plants. And the Rhamphicarpa free plants are those with the open symbols, and the infected plants are those with the gray or the black symbols. So you see that at 41 days and at 49 days after sowing, there are no significant effects yet. And gradually over time, you can see that the effects are becoming more and more important. So indeed, infection of Rhamphicarpa steeply reduces the host photosynthesis, but also the leaf chlorophyll content and other leaf physiology parameters. But the effects that we observe, they start much later than with Striga. So in Striga, we've seen that these kind of effects they appear to appear much earlier. We also observe that the parasite first extracts assimilates before it really impacts the host photosynthesis and growth. So there is, if you want, there is a little bit of a delay there. Now, in terms of how the host influences the parasite, we have observed that the Rhamphicarpa fistulosa seeds, they are short-lived, and they also need light to germinate. So this is also quite a distinction from Striga. Striga has very long-lived seeds, and they don't need light. They can germinate from the soil. Better yet, they need to be in the soil because they need to be close to host roots. So the Rhamphicarpa fistulosa seeds, they can germinate just as ordinary weeds. And this has implications for management. So they could, for instance, be controlled by deep tillage, by burying the seeds into deeper soil layers. And if they're not exposed to light, they will not germinate. We've also seen, very importantly, that Rhamphicarpa seed germination is independent of host presence. And this is shown in this figure On the top figure, you can see Striga hermonthica, and on the bottom figure is Rhamphicarpa fistulosa. With Striga, you can see that GR24, which is a synthetic, well, it is a strigalactone analog. It is a germination stimulant for parasitic plant species. And if we expose these seeds to 10% or even 1% of GR24, you see that there's a lot of Striga, a high Striga germination percentage with that germination stimulant. We have also exposed these Striga seeds to root exudates of different rice varieties, and of course these root exudates also contain the strigalactones that stimulate the germination of Striga. And we see, although lower than with GR24, but we also see germination with rice root exudates. But if you only apply water, you can see with Striga hermonthica, you don't even see any germination with water. Now, if we go to the Rhamphicarpa seeds on the lower part of this, the second figure, you see for all these type of treatments, the Rhamphicarpa fistulosa germination is very similar. So there are no effects of germination stimulants. There are no effects of the presence of a host plant. And this, again, has implications for management. For instance, crop rotations, which is really effective against Striga. Crop rotations or fallow, not growing any crop for some period of time, that is effective for Striga, but may not be effective for, or not as effective for Rhamphicarpa. Now, based on all this work on the host effects and the parasite-host interactions, we could draw a timeline for both the parasite and for the host. Now, let me start with the parasite. After sowing, around 6 days after sowing, you can expect germination and emergence of the parasite. Then around 36 days, we can see maximum germination and really the start of parasitism. So it takes quite some time before they start to parasitize. Then you first need to grow a plant. They need to grow roots, and then the roots need to find roots of a host plant. And then after some time, at 57 days after sowing, the biomass the biomass of parasitizing plants is really gaining from parasitism compared to non-parasitizing plants. At 70 days after sowing, there's flowering, and at 100 days after sowing, the seeds are maturing or are mature, depending on when they started to flower, of course. And for the rice, the changes in root shoot biomass allocation, they start a bit later than the start of the parasitism. And then gradually you get reduced light interception, you get arrested biomass production, you see significant effects on host photosynthesis, biomass, leaf area, and tiller number losses. And then it flowers and it matures, and all of this much later than the parasites, which is important. So based on this, we identify 3 different control windows. The first control window is in the period before the maximum germination, before the start of the parasitism. This is if you control the weed in here, you can really you can really prevent parasitism. But even if you are too late, if farmers would be too late, they could still weed the parasite and by that prevent the damage. So there's a second control window. They can actually prevent a lot of crop damage when they do the weeding, even if it is after 36 days after sowing. And then there is actually a third control window, which would be beneficial on the longer term, if they would be able to remove or kill the parasites, the parasitic weeds, by the time they get into the generative stage, when they're flowering or producing seeds. Then they can also prevent seed bank replenishment. Now, talking about control, we have also investigated different means of control. So the first thing that we did was we were looking at the rice varieties. are there any differences in resistance and tolerance between rice varieties? So we have grown 64 different rice varieties in this field. You can see it on the picture. And we saw that within this group of rice varieties over 3 different years, we saw yield loss ranging from 24 to 73%, and the mean yield loss by Rhamphicarpa is 50%. And so here on the top picture, the green boxes, they show Rhamphicarpa fistulosa biomass for different varieties. And there you can really see that there are some rice varieties like Nerica, lowland Nerica 3, lowland Nerica 23, and also the lowland Nerica parent IR64. They are actually quite resistant. They generate, or the Rhamphicarpa fistulosa plants generate much lower amounts of biomass on these varieties. We also see tolerant varieties. So on the lower hand, on the lower graph, are the relative yield losses for the different varieties caused by parasitic weed infection. And there you see like the local variety Supa India, but also the improved variety, lowland Nerica 3, they have really much reduced relative yield losses compared to some of the other varieties. So that is already really good to be able to recommend to farmers. Then we also looked at sowing dates. Does it matter at what time you are sowing the crop? And we found out that it does indeed. With lower biomass observed with earlier sowing, you see that on the left-hand side of the graph, the green boxes. And then this also, and this is opposite of what we observe with Striga asiatica, interesting enough. There we see that early sowing is actually better than late sowing. And we also see that the rice yields are generally higher with early sowing. And this finding will allow farmers to maintain their preferred variety, which is late maturing, at least in Tanzania. So Supa India could be recommended because that is a late maturing variety that is highly preferred by farmers. And for those farmers in Tanzania, it proved really difficult to make them enthusiastic about any other types of rice varieties. They are really fond of their Supa India. So this is actually a really tangible and good outcome for them. Then we also looked at soil amendments. Can soil amendments be a control measure? And we looked at different types of soil amendments. We looked at mineral fertilizers, NPK and DAP, but we also looked at locally available organic fertilizers like cattle manure, rice husks, and we had also treatments where we had half of the recommended rate of NPK or DAP, of diammonium phosphate, and then supplemented by any of the organic fertilizers, cattle manure or rice husk. And surprisingly, we saw that the Rhamphicarpa fistulosa does much better, or does quite a bit better when it is fertilized than when it is not fertilized, when the crop is not fertilized. This is opposite of what you see with Striga asiatica or with any other Striga species. There you really see fertilization is really a good control measure. So this is not the case for Rhamphicarpa. But still, the rice, and you see that on this in this box plot, you see that the rice grain yields, despite infection, are still benefiting from these different fertilizer applications. And then particularly the rice husk, and the rice husks also improve soil fertility. I'm not showing that here, but it is shown in the paper. So based on this, we could advise farmers to replace at least part of the mineral fertilizers by rice husks, so that they do not waste too much of the fertilizer, because a lot of the fertilizer will go actually to the parasitic weed. But if they do that, they should still combine it with timely weeding, because like I said, they're also fertilizing the wheat, and that is not what you want. But in terms of the rice yields and in terms of soil fertility, rice husk can play a really good role there. Then we were intrigued by this finding, and we wanted to dig a little deeper in what does nutrient availability actually do for both the host and the parasite. So what effects do fertilizers have on infected host plants, and also what effect do fertilizers have on the parasite? So we conducted a number of pot experiments, and we are actually still working on this. We do currently more pot experiments with this on this species. But what we have found so far is that here on the y-axis you see the above ground biomass of rice plants, and on the x-axis you see the different levels of fertilizer, so percentage of the recommended rate, and you see that the parasite-free host, the black dots, they are quite responsive. They're very responsive to the fertilizer levels, of course, what you expect. And then the open symbols on the let's say the lower dots, the lower points, they represent the parasite-infested hosts. So you see that the host hardly gains from nutrient application if it is infested by the host. And then in the middle line, the axis, that shows the combined biomass of the parasite and the host. So interestingly, you see that the parasite-free host biomass, so the black dots, they're always higher than the total biomass of the parasite and the host combined. And this is actually quite typical for parasitic weeds, because they have a sort of a phytotoxic effect. They're not just, it's not just a sink-source relation. There's also a physiological effect that is playing a role, and that causes actually that the combined biomass is lower than the biomass of healthy plants. This is a picture of the pots, and you see the different fertilizer treatments, and you clearly see the first pot in every picture on the left-hand side is the parasite only, and you really see that the parasite only is not affected by the fertilizer. Oh, I should actually show that on the next slide. But here you see that the middle pots where the rice and the Rhamphicarpa are combined, you see that the total biomass is lower than what you see on the right-hand side where the rice is growing alone. Now we have looked at the parasite, and I already said what I should be saying right now, and that is that when the parasite is growing alone, and those are the black dots, so without a host, there is no response of the parasites to the fertilizers. So on the upper graph you see the Rhamphicarpa biomass, and on the lower graph you see the Rhamphicarpa capsule dry weight, and they respond in a similar way. When they are actually with a host, so when the Rhamphicarpa is growing, parasitizing a host, then you see all of a sudden a fertilizer response. So parasitizing Rhamphicarpa fistulosa benefits from increased nutrient availability, and host-free Rhamphicarpa plants do not benefit. So that is quite an interesting result. And then we also looked at what is happening below ground, and there you see for the parasite on the right-hand side, you see exactly what you also see with the above ground biomass, no response for the parasite without a host, and steep fertilizer response when it is growing with a host. And also on the host, so the left-hand side of these two graphs, for the host you can see that the parasite-free plants are responding to fertilizer increases, but also the parasite infested ones, but the parasite-free ones are responding better. So what you can see from here, what you conclude from here, that is when the parasite grow with a host, not only the parasite itself can grow more roots, but it also can gain from the increased root and carrying capacity of the host. And that is why you see here on this picture again, this is at physiological maturity. You see the left-hand side pots, you see the parasite growing alone, you see no effect of the fertilizer. But when it is growing with a host, the middle pots, then you can see that it is responding on the responding to the fertilizer, to the increased fertilizer dosage. So conclusions, and I'm sorry that I took longer of your time than expected. Conclusions: rice vampireweed is a widely distributed and increasing weed of rain-fed lowland rice. It can co-occur with Striga, but it prefers wetter soils. It is not dependent on host presence, and this is very unlike with Striga, so with obligate hemiparasites. But the reproductive output of Rhamphicarpa and also the fertilizer use efficiency, they both strongly benefit from a host presence. It affects the host in a similar way as Striga, but quite a bit later, so there's quite a bit of a delay. And actually, this is not, this is difficult to prove, but my observation is that it is also deadlier. It is on the plant level, Rhamphicarpa has an even worse effect than Striga. The yield losses range from 24 to 73%. But fortunately, we have also identified a number of control options. They can be controlled by resistant and tolerant rice varieties. They can be controlled by earlier sowing. As a sideline, Striga is controlled by late sowing. So there's again a different response here. But also, Rhamphicarpa could be controlled by ordinary weed control methods. And you actually have quite a window of opportunity there, because there is a delay in when they start to parasitize on the crop. So that is the period where you can ideally control the weed. It cannot be controlled by fertilizers. And this is also, again, opposite from what we found with Striga. But we can still recommend fertilizers to be used by farmers, because the fertilizers can mitigate some of the negative effects of the parasite on the crop. So this work is done with a lot of people, of course. My main collaborators are Lambert Bastiaans from Wageningen University, the two students that we co-supervised, Stella Kabiri from Uganda, Dennis Tipper from Tanzania, my former postdoc Mamadou Sissoko from Côte d'Ivoire, and local collaborators, Galbert Benuru from Benin and Juma Kayeke from Tanzania, and one of the early students, MSc students with whom I worked with in Benin, Norliet Sosu Kudering. And these are the organizations that were involved, and the logos are there as well. And a lot of this work has been made possible by funding by the NWO WOTRO Science for Global Development. That's the Netherlands Foundation of Scientific Research. I would like to thank you for your attention and also for inviting me for this talk.